Willets from the University of Texas at Austin.
All right, let's try that. So it is my extremely great pleasure to be here today to help honor WE for his receiving the Pittsburgh Spectroscopy Award. And I'm also extremely excited because WE did a lot of the heavy lifting for me. And so I don't have to go through and explain some of the concepts of super resolution imaging in as great detail. So what I'm going to talk to you about today is how we're going to use techniques in single molecule spectroscopy in order to study metal nanoparticle hotspots. And so as WE introduced, the idea of doing single molecule spectroscopy has many powerful advantages, one of which is that we can reveal heterogeneity in complex systems. And we just saw a number of examples in which biological systems, which have an inherent heterogeneity, were studied using this approach. What I'm going to talk to you about today is an extremely heterogeneous system based on noble metal nanoparticles that support localized surface plasmons and lead to a phenomenon known as surface-enhanced Raman scattering, or SIRS. And so the basic idea of SIRS is that we're going to take a molecule of interest and adsorb it to some sort of nanoscale featured metallic surface. And for most of the data I'll be talking about today, the featured surface of interest is going to be a metallic nanoparticle made out of silver. And so when we take an analyte, such as this rhodamine 6G molecule shown here, and allow it to adsorb to this metallic nanoparticle structure, what we find is that the Raman scattering is enhanced by many orders of magnitude, typically 10 to the 6th to 10 to the 8th. And since Raman scattering is such a weak process, only 1 in 10 to the 8th photons that are scattered by a molecule will be inelastically or Raman scattered. The ability to have a signal enhancement on the order of 10 to the 6th to 10 to the 8th is a very strong advantage. So the reason this works is because we excite localized surface plasmons in our metallic nanoparticles. And the basic idea is that the electric field of our light is going to pass by our structure, and because the nanoparticle is smaller than the wavelength of light, all of the surface conduction electrons will be displaced at the same time. And so we end up with this collective displacement of the surface conduction electrons as that light wave propagates over our nanoparticle. And this is the basic principle behind a plasmon. One of the reasons we're particularly interested in these plasmons is that they lead to very strong electromagnetic field enhancements at the surface of the metal nanoparticle. So in the case of an isolated metallic sphere, such as shown here, when it's excited with light polarized in the vertical direction, we get electromagnetic field enhancements that are 20 times higher than the incident electromagnetic field at the surface. On the other hand, when we have pairs of nanoparticles, such as the dimers shown here, illuminated with light polarized in this horizontal direction, these electromagnetic field enhancements can be even higher. And the intensity in the gap here will be enhanced by a factor of over 10,000. So this is extremely powerful, and this is the basic principle behind what we call an electromagnetic hotspot. So in the case of these hotspots, there are very special circumstances when the electromagnetic enhancement is sufficiently high to produce SIRS from a single molecule. So here are 4 examples shown from Richard Van Dyne's lab, where they found these silver clusters which basically had these gap-type structures that should generate those strong electromagnetic field enhancements and produce single-molecule SIRS. And you'll notice there's a strong diversity in the shape and structure of these different nanoparticles, bringing us back to this concept of local heterogeneity, which is really going to be important for single-molecule studies. So when we look at this, we have several questions that emerge from these type of single-molecule SIRS data. One is, are the hotspots actually located in the junction as theory predicts? To date, most of the data has simply shown that the nanoparticles that support SIRS from a single molecule are in fact nanoparticle aggregates, indicating that the junction should be responsible for this electromagnetic field enhancement. But verifying this is extremely challenging experimentally. Other questions include, are all junctions single molecule SIRS active? So for example, in this trimer shown in the lower left, you can see that there are 3 possible hot junctions. And we have no way of knowing whether all of these junctions are hot or whether there's something special going on the surface of these nanoparticles. And lastly, you'll notice these aren't very homogeneous. And it turns out it's extremely difficult to generate single molecule SIRS from a homogeneous nanoparticle sample. For some reason, it's really these sort of, I call them dirty nanoparticles, because they basically are just very heterogeneous in shape and size. These seem to be the best substrates for supporting single molecule SIRS. So what we'd like to do is address these questions, but as we learned from the last talk, we run into this fundamental limit called the diffraction limit of light. And even though there's a huge size mismatch between the molecules on the surface of the nanoparticles, due to the fact that the molecules are on the order of 1 nanometer in size, whereas the nanoparticles are on the order of 100 nanometers in size, we see that when we image them in a standard far-field optical microscope, they basically look indistinguishable, whether we're looking at 1 molecule, 1 nanoparticle, a single nanoparticle decorated with many molecules, or several nanoparticles decorated with 1 molecule. They all look like a diffraction-limited spot or blob. And so this is really the goal of our study, is to apply some of these super-resolution imaging techniques that we've heard about from WE's talk and try to defeat the optical diffraction limit. So for all of the data I'm going to tell you about today, this is the basic sample preparation we'll use. We'll start out by preparing what are known as the Lee and Mizell silver colloid. Again, these are these dirty silver nanoparticles that are just made by reducing silver salt in the presence of sodium citrate. To that solution, we'll then add our SIRS tag, in this case rhodamine 6G, at a concentration such that we expect to have roughly 0 to 10 molecules per nanoparticle. So we're down close to the single molecule limit. And then we'll add either sodium chloride or sodium bromide to facilitate nanoparticle aggregation and create these junction regions, which we believe to be the hot spots. This solution will then be drop cast onto a glass cover slip and imaged in an optical microscope like the one shown here. Basic ideas will come in with 532 nanometer laser excitation and epi illumination, pass that through a high numerical aperture microscope objective, collect the resulting SIRS scattering, and pass it to one of 2 detectors. The first detector is going to be one of these fast electron multiplied CCD cameras. And this is what we're going to use to generate SIRS images. And so we'll get a series of diffraction-limited spots in a movie like the one shown here. The other detector is a spectrometer coupled to a liquid nitrogen-cooled CCD camera. And this will allow us to measure SIRS spectra. So once we have a series of SIRS images, we can then process them using this point spread function fitting approach, or part of this super localization microscopy that WE described in detail earlier. For our data, we're going to take each diffraction limited spot and we're going to fit it as a 2-dimensional Gaussian. So we'll approximate this data, the raw data, as this 2D Gaussian and ultimately extract the parameters x0 and y0, which correspond to the position of the emitter. As WE mentioned in his previous talk, if we want to resolve individual emitters within a single diffraction-limited spot, we need some way to modulate the signal. And he talked about the idea of using active control based on triggering a molecule between an emissive and non-emissive state. In single molecule SIRS, it turns out that the molecule does this for us. We don't have to put any work in. And so here I'm showing just a time series of Raman spectra where you can see initially we observe no Raman data. Then our molecule turns on, then it turns off again, comes back on, etc. So these native intensity fluctuations are going to be extremely helpful for us in terms of our ability to analyze these data using super resolution approaches. And it turns out we need to use super resolution microscopy because we do in fact have 2 emissive sources on at the same time. When our SIRS is on, we're going to observe a very bright image as shown here. But also in that same image is a very dim but present background luminescence coming from the silver nanoparticle itself. And so what we need is a way to resolve these 2 emission sources, this nanoparticle luminescence, as well as the SIRS coming from our molecule of interest. So the way we do this is we're going to look at the intensity of our diffraction-limited spot as a function of time to create a time trace like the one shown here. During the high intensity times, we know that our SIRS is coming from our molecule of interest on the surface. However, after some period of time, the SIRS ceases in a single digital step, most likely due to photobleaching of the molecule on the surface, although it could also just migrate out of the hot spot and no longer generate single molecule SIRS signal. we define an intensity threshold below which we only have the nanoparticle luminescence, and this corresponds to frames or images that look like this. We'll then apply this point spread function fitting approach by fitting to a two-dimensional Gaussian, and we'll extract x naught and y naught for all of those frames. And that's what's shown here as the black data in this scatter plot. And so these black points represent the centroid of our stable background luminescence coming from the silver itself. And what we notice about these data is that they're symmetric around the mean. And we would expect for a stable emitter for us to basically fit to the same location every time if it's coming from the same location. But because we have some small amount of noise in the system, we observe scatter in this data with a standard deviation of 6 nanometers. And using the equation that WE showed in his previous talk, we can actually confirm, based on the number of photons we have, the pixel size of our detector, and other experimental parameters, that we would expect this standard deviation to be on the order of 5 to 6 nanometers. So this agrees very well with our expectations from theory. What we then do is we turn to the frames above this intensity threshold when our SIRS emission is on. We subtract away the point spread function contribution coming from that nanoparticle and are left behind with just signal coming from the SIRS emission itself. We then fit this to our two-dimensional Gaussian, and we get the centroid positions that are shown here in red. And these represent the centroid of our time-varying SIRS signal. That is, each of the locations corresponding to all of the data points above this threshold here. Now, the first thing we notice when we look at these data is that we have an ellipsoidal shape, and it extends over 40 nanometers in length here. For these data, based on our number of photons, we would expect to have resolution on the order of 1 nanometer. So clearly, this is extending much further than our predicted or expected resolution. Lastly, we notice that the SIRS data is offset from the center location of our luminescent centroid. So because we have so many points here, this is not a very useful way of looking at these data. So what we're going to do is we're going to create a series of histograms. So here, just for convenience, I'm representing the luminescence as a white X, and then I'm showing all of the red scatter points corresponding to our SIRS emission. First, we're going to create what we call a position histogram, where we're simply going to create bins across these data, just creating a grid over this data, and count the number of fits we get in a particular bin. And so this is the result shown here. And again, we see we preserve the elliptical nature of the data, and we still see this extended length over 40 nanometers. So we know that this isn't just noise in the data, but it's a real phenomenon. The next thing that we do is we're going to create something called a spatial intensity map. And the idea here is we're going to look at each bin within our histogram, for example, this bin shown here, which has 40 associated data points, and we're going to calculate the associated SIRS intensity of all of the points within that bin. And this is the result shown here. So again, the color scale here is showing the intensity of the SIRS as a function of the position in X and Y. And there are immediately several trends that are apparent within these data. First and foremost, we see that the SIRS intensity is highest at this upper right corner, close to our luminescent centroid. And then we see it decays away with a gradient fashion with strong directionality. Second, that we still preserve that size dimension that I mentioned earlier. This is extending over 40 nanometers. And previously in the single molecule SIRS community, researchers had hypothesized that the size of the hotspot was on the order of a single molecule, on the order of 1 nanometer in size. There's a hole right there. That was really scary. So in this case, we are seeing that we're observing the intensity extend over a distance much larger than the size of a single molecule. Lastly, we can actually plot these data as a diffusion trajectory. So here I'm showing now the X dimension, the Y dimension, and the time dimension, and the color scale corresponds to the SIRS intensity. And we actually see that the molecule is exploring a smooth trajectory towards this region of high intensity and then away from that region. So this isn't just stochastic data. There's actually a time dimension to these data. This is not an isolated event. Here's another example. Now the molecule is actually turning on and off over multiple cycles, so we're seeing the intensity go high, then low, high, then low. Again, we can create one of our spatial intensity maps, and we can show both the site of the nanoparticle luminescence, marked with a white X, as well as the region of highest SIRS enhancement that then decays away in a gradient and directional fashion. If again we plot these out as diffusion trajectories, I'm showing this now for the 3 points labeled 1, 2, and 3. what we find is that, again, we have a smooth, time-dependent motion in and out of the hotspot. So in the first one, we start here. We go in towards the hotspot, or the region of highest intensity, before moving out. In the second place, again, in, then out. And in the third example, in, then out. So we're having this recurring excursion towards this same region that corresponds to the highest overall intensity. So to understand this, we actually turned back to some theoretical calculations done by George Schatz's group. Because initially we thought, well, we must be looking at the motion of the molecule on the surface of the nanoparticle. But that's a naive expectation simply because we must recognize, in the case of SIRS, the molecule isn't radiating out directly into the far field, but is instead coupled into a plasmonic nanoparticle. So if we think now about the SIRS enhancement, we have two primary enhancement sources. SIRS is typically referred to as an e to the fourth effect as a result. The first enhancement results from plasmonic excitation due to the excitation field itself. And this is the typical type of example that you see here, where you have a plane wave exciting a plasmon in the nanoparticle, and you get a locally enhanced electromagnetic field on the nanoparticle surface. Now, this can excite the SIRS in the molecule, but when the molecule emits back down, it's going to couple back into the plasmon. But it's no longer going to behave as a plane wave coupling into the plasmon, but instead a point source coupling into the plasmon. And so this is going to be our Raman scattered light, this second term here. And calculations from Logan Ausman and George Schatz show us that if our dipole, or our point source, is sitting in the junction between our two nanoparticles, we would expect the light to be distributed or re-radiated by those nanoparticles evenly over the entire nanoparticle structure with extremely high intensity, because you have strong coupling between your point source and your plasmon. On the other hand, if our dipole is sitting more on this leftmost nanoparticle, it's going to couple into the plasmon associated with this leftmost particle more strongly. And that's going to bias the emitted light, or scattered light, towards this leftmost particle. So we'll see a shift in the center of mass of our scattered light. But we'll also note that the intensity scattered by the nanoparticle is less compared to this case over here, simply due to the fact that you're not coupling as efficiently to the plasmon. So this gives us an idea of explaining what we're observing experimentally, where if we have our dipole very close to this center region here, close to the hotspot, we expect to get very strong SIRS, and as that molecule moves away and couples less strongly to the plasmon, we observe a corresponding decrease in intensity and shift in the centroid of the emission. On the other hand, the metaluminescence, we believe, models the overall plasmon enhancement of the nanostructure. So it's not coupling in as a point source, but we can instead use this plane wave approximation. And therefore, we would expect in the metaluminescence case to see that luminescence associated with that center region. And for this reason, we think we have good overlap between the site of our metaluminescence and the site of our strongest SIRS. So this mobile dipole on the surface really helps us to explain this relationship between why we see an intensity-dependent spatial relationship between the SIRS that we measure and its emission location. However, there are several examples where we do not see the hottest SIRS overlap with our metaluminescence. So we have this question as to why these two data don't overlap, and we suspect it has to do something with the structure of the underlying nanoparticle. So what we'd like to do is to relate these spatial intensity maps to this underlying structure, and ideally understand why the SIRS and the luminescent centroids sometimes show offsets. So to do this, we're going to perform correlated optical and electron microscopy studies, where we're going to use patterned ITO cover glass, which is both conductive and transparent. Conductivity is important for doing the electron microscopy. Transparency is important for the optical microscopy. Then we can go into our optical microscope and image this slide under both dark field and laser excitation, and then pattern matched with our electron microscopy results. So just to give you an idea of answering this first question, does the spatial intensity map reflect the geometry of the underlying nanoparticles? We'll start with this spatial intensity map shown here, where we observe the strongest SIRS along this edge that decays in gradient and directional fashion. The correlated electron microscopy image is shown here, and in this case we have a nanoparticle dimer, which is nice because it gives us the ability to relate this back to theory. From our theoretical expectations, we expect the highest intensity to be associated with a dipole sitting along this nanoparticle junction, and then the intensity to decay away as we move away from that junction. What we find is we have excellent qualitative agreement with the geometry and orientation of this high-intensity edge in our spatial intensity map and the geometry and orientation of this junction region between the two nanoparticles. So if we simply resize the spatial intensity map and place it on top of the electron microscopy image, we find that we get very good qualitative agreement based on our expectations from theory. As a second example, I show this spatial intensity map, where again we have the strongest SIRS along this edge that decays away in gradient and directional fashion. If we look at the associated nanoparticle structure, we find that we have two possible hot junctions, one of which is oriented vertically, which doesn't agree with the orientation of our hotspot map. On the other hand, the orientation of this junction shown here agrees very well with the orientation of our high-intensity edge in our spatial intensity map. And again, if we resize this image and overlay them, we have very good qualitative agreement between the spatial intensity map and a structural feature of our underlying nanoparticle. So we feel, based on these data, that this is good evidence that we do in fact have a junction-mediated SIRS emission phenomenon. But what we'd like to do is to have a more quantitative way of assigning these two relationships. And for this, we're going to exploit the nanoparticle luminescence that's inherent to the nanoparticle itself. So to illustrate this idea, I'm showing now a third example of a spatial intensity map, this time with the associated luminescence centroid that is now offset by several tens of nanometers. In this case, the correlated electron microscopy image shows that we have a nanoparticle trimer. And so just based on a qualitative assessment, we would say, well, this is a horizontally oriented map. I have a nice horizontally oriented junction here. So let's toss it on there and see how that goes. And this agrees qualitatively with our expectations, but what we'd like to do now is to understand why we're seeing the luminescence emerging from a leftmost biased nanoparticle shown here. So to do this, we're going to just employ discrete dipole approximation calculations in collaboration with David Masiello at the University of Washington and his graduate student, Jonathan Litz. And what we did in these calculations is we said, okay, we know that the electromagnetic field at the surface of the nanoparticle will be enhanced relative to the incident field. So we're going to calculate the centroid, this value x here, based on a weighted, an intensity-weighted position. And we'll do this by weighting it either as an E squared, so an intensity weighting, or we can do this as an E to the fourth, which models our SIRS, so an intensity squared. And the idea is that we're speculating that this E to the fourth centroid should model our SIRS position very well, because as I mentioned, SIRS is an E to the fourth electromagnetic field enhanced mechanism. And we hypothesized that an E squared enhancement would better represent this underlying metal luminescence. And I want to point out here that this analysis is not accounting for the presence of the molecule. So we're not actually putting a dipole on and looking at this re-radiation. This is just a plasmon analysis. So if we do the calculation, we get the following results. We find that both the E squared and the E fourth centroids are localized within, less than 10 nanometers of each other, and they're both located on this leftmost nanoparticle, consistent with where we've placed our luminescence data. So in terms of agreement with our luminescence centroid, we have excellent agreement between theory and experiment. But we notice that the SIRS data is not showing up in the location that we would predict based on our experimental assignment. However, this led us to hypothesize that the silver luminescence is probing a collective plasmon enhancement averaged over the entire nanoparticle structure. But in the case of SIRS, our molecule is very small and is only going to be sampling a local plasmon mode based on its position or location on the nanoparticle surface. So in this case, if we hypothesize that the molecule is actually sitting in this junction here, it should efficiently couple into this plasmon mode associated with that junction and not couple very efficiently into any other plasmon modes on our nanostructure. So if we redo the calculation under that assumption, what we find is that our E to the fourth centroid will actually shift to that junction as expected. And if we just compare now our E squared centroid, which is now averaged over the entire nanostructure, versus E to the fourth, which is only averaged over the plasmon excited locally, we find that we get excellent agreement between our theoretical predictions and our experimental assignment. To further validate this model, I'm going to show you 3 dimer examples. And these dimers have different relative orientations with respect to our excitation polarization. In the first case, we see that our hot sites in our spatial intensity map are strongly co-localized with our luminescent centroid. In the second example, where the dimer is now oriented at about 45 degrees relative to the excitation polarization, we see a small offset between our strongest SIRS and our luminescence. And in the third example, where our nanoparticle is now orthogonal to the excitation field, we see a very large difference between our SIRS centroid and our luminescent centroid. So if we go back to this idea of plasmon enhancement, again, we're hypothesizing that the molecule is sitting in this junction between the nanoparticles, and we've placed our SIRS hotspots accordingly. Now we have to think about why we would expect to see the silver luminescence showing up at the position where it does. So for this, we turn to calculations from Ube and Nordlander on asymmetric dimers. And so in this case here, they have an asymmetric dimer where the light exciting it is polarized along the long axis of the dimer, which is similar to this example shown here on the left. What we find is that the strongest plasmon enhancement occurs in the gap here. And if our luminescence is tracking with that plasmonic enhancement, we would then expect to see our luminescent centroid strongly associated with the junction, which is consistent with what we've observed experimentally in this leftmost nanoparticle example. On the other hand, if we have an asymmetric dimer oriented orthogonal to the excitation field, now we're going to see that one nanoparticle is going to be excited preferentially relative to the other. This should conceivably shift the plasmon enhancement up towards one of those nanoparticles because it's being excited more strongly. In the case of this asymmetric dimer shown here, we expect that the light is actually exciting the plasmon associated with the lower nanoparticle more strongly than the plasmon associated with this upper nanoparticle. As a result, because our silver luminescence is tracking with this collective plasmon enhancement, we expect to see our silver luminescent centroid shifted down towards this lower nanoparticle as we observe experimentally. So we think we actually have a really nice handle on why the silver can be offset from our SIRS centroids. To verify this, we once again turn to our DDA calculations with the Masiello group. And we were only able to get good results for these 2 structures shown here. Our third example was a little too computationally expensive for the DDA method. But we do observe that we get excellent agreement between our calculated SIRS centroids for both e to the 4th for SIRS and e squared for the luminescence with our experimentally derived results. What we also note is that we actually observe better agreement between our e squared centroid and the silver luminescence, supporting our idea that this luminescence is better described as an e squared enhancement mechanism than an e to the 4th enhancement mechanism. We also note that the e to the 4th centroid agrees very well with the average SIRS centroid. We recognize once again that the SIRS is really telling us where the molecule is on the surface and how well it's coupling into this plasmon mode. And so we don't expect for this SIRS centroid to simply converge to a single location, but rather to wander about on the surface due to molecular motion. Once again, our data supports this idea of a junction-mediated single-molecule SIRS emission. So if we now put it all together, this is where we've come. This is sort of where we are in the story at this point. So far we've shown that super-resolution imaging shows us that there's this relationship between the spatial origin of our SIRS emission and the intensity of the SIRS we see. We found that the shape of the SIRS spatial intensity map show qualitative agreement with the nanoparticle structure and geometry and seem to agree with the idea that SIRS is in fact a junction-mediated phenomenon. We've also shown that SIRS is a local plasmon effect that's dominated by the junction in which we find the actual molecule. It's not coupling to the entire nanostructure, but instead to a local plasmon mode. On the other hand, this silver luminescence seems to be collectively enhanced by the overall plasmon of the entire nanostructure. And lastly, the important part is that we wouldn't be able to access this information without single molecule spectroscopy. We really truly have a heterogeneous system here that requires the ability to go down to the single molecule level. And for that, we owe a debt to this man right here. Sorry, WE. This is his caliper. Because he was really one of the first people to show us that single molecule imaging was possible. And so none of this work would really be possible without having his initial experiments. Of course, I need to acknowledge the students that have actually done this work. And so for that, I have to acknowledge Sarah Stranahan, shown here, and Maggie Weber, as well as our collaborators, David Masiello and Jonathan Litz at the University of Washington, and you for your attention. I'd be happy to take some questions.
Thank you, Kelly. In this case, we do have a few minutes before the next talk is due to start. Does anyone have any questions?
So we've done a little bit of that to try to really map out, as we rotate the excitation field, how we see the centroid move. We've seen a little bit of motion, but we really run into photobleaching. So photobleaching is our big enemy in this case. So we haven't gotten a really nice correlation between the two, but we know there is some correlation. So we're still working to map that out. It's a great question. Julie? There is absolutely a wavelength dependence. So we've also been working on that. And one of the major challenges we have with the single molecule data that we're finding is a nanoparticle that's really good at 532 nanometer excitation doesn't work as well if we go on either side of it. So we'll hit it with either 488 or 642, and we're not getting the same single molecule SIRS signal out. So we're actually now working at the, don't hurt me, WE, the ensemble limit. So we're increasing, we're going up to having more molecules on the surface. The idea being that the SIRS should still be coupled into different plasmon modes when it's being emitted. And so we're hoping that we'll be able to see a shift between the two as we work at that limit. Because you're absolutely right, the wavelength dependence should be there. Randy. So does the charge matter? I think it does, but we're still working on that. We have tried a few different dye molecules to explore that. One of the challenges is that there's sort of two intensity states that we tend to find. We tend to find very rapid fluctuations in intensity, and occasionally we get ones that will stay on long enough that we can fit that to a diffusion analysis. So yes, we see a intensity dependence in terms of the diffusion coefficient that we measure. We also see what's termed as subdiffusion. So we actually don't see Brownian motion of these species on the surface. We're getting this sort of very strange subdiffusive behavior, and there's actually two subdiffusive regimes. So we're trying to get to the bottom of this right now, but it's interesting, but it's tough because getting the data to actually behave so we can analyze it has been a problem. WE. Yes. But in order to observe that, you have to have certain turning on and off. What's making it turn on? We think it's that the molecule's actually diffusing out of the hot zone. So you're getting to a point where you're no longer enhanced enough to observe signal above the background. That's our hypothesis. But what's interesting is that, if you go back to some of these time traces, you really see this sort of step up to high intensity, step down to low intensity. We do see a little bit of a trajectory involved in it, but one has to wonder if there's really this off-on point on the surface. So I don't know the answer to that. I think we're still really trying to figure this out. The other variable that we just don't have a good handle on is orientation of the molecule. And that's a real challenge because it's been predicted that if a molecule is oriented along the long axis of the junction, you should have very efficient coupling, whereas if the molecule becomes orthogonal to the junction, that coupling should basically go off. And so that's another intensity effect that we just can't really get to because there isn't going to be a corresponding polarization like in the case of a single dipole in fluorescence. Because in our case, our dipole is coupled into the plasmon. The plasmon mode actually dictates the output polarization, which we've shown through a couple of recent papers. That's a good question. So we measure the surface plasmon resonance before and after to make sure that we're not blasting the particles to pieces because that would be bad. So if we do see a change in the plasmon resonance, we know that that means that we've done laser damage. We're typically working anywhere from 0.7 to 7 kilowatts per square centimeter. That's typically the range in which we operate.
I'd like to thank Kelly one more time. And our next speaker is Julie Bettine from the University of Michigan.